Now that we understand a little bit more about DNA and protein synthesis, we want to come back to some of those questions we asked at the beginning, whether or not you are okay with you or your children eating genetically modified foods, or whether you agree with cloning or things like that. Well, first off, about 85% of United States corn and soy are genetically modified. Other processed foods that contain genetically modified materials would be like corn syrup, soy, vegetable oils, grape-flavored drink, grapes themselves, genetically modified. And so we also see what we call transgenic organisms, where you take genes from one organism, you put it in another. For example, we can take bioluminescent fish, and we can put those genes that make them glow into pigs and make them glow as well. So why in the world is this important? Why would we want to genetically modify plants and animals, besides the fact that we can make them glow in the dark and things like that? Well, for plants, it can increase their shelf life. The way that they are genetically modifying tomatoes right now, it makes them ripen more slowly. So as far as grocery stores are concerned, they're big fans of this because they can purchase tomatoes, they can put them in their store, and they're going to have a longer shelf life. So they're going to be fresh longer. They're not going to rot as fast. It's also good for the consumer because that means you can buy tomatoes and you don't have to hurry up and eat them all really fast because they're going to last longer. We also know that modifying plants can increase crop yield because we can modify plants in such a way that they resist pesticides. Herbicides don't harm them, and so you can use those pesticides to kill the bugs, but it doesn't kill the plants. So that's a good thing. It also can increase nutritional value. So we can produce golden rice with beta-carotene in it. We can take potatoes and we can genetically modify them in such a way that they actually give us materials, vitamins and minerals that we need that we may not normally be getting in those foods. Now, there are some concerns. We do believe that genetically modified crops can cause the pests around them to become resistant, and so this can develop superbugs or super weeds that pesticides and herbicides are not able to kill. So that's a problem that we could see with them. Now, as far as modifying fish, so we're actually finding what we call transgenic salmon, which carry other fish genes. So we're taking genes from other fish, putting them in salmon. It's causing the salmon to grow larger, causing them to grow faster than normal. And so, that's a good thing for them. Fertilized Alaskan salmon eggs are actually injected with growth factor genes to make them grow larger and faster. One I guess, problem or concern with this is that these salmon in the lab are typically sterile, and so that's why they're not released in the wild. But the concern would be, what if these engineered salmon are able to mate with wild populations, and it could totally disrupt food webs. It could totally disrupt a lot of different species that are found in those habitats. Another thing that we know about genetically modified plants and animals is something called farming. What farming here does is it produces genetically modified organisms that produce drugs to treat or to prevent disease. So it actually blends pharmaceuticals, drugs, with farming. So for example, tobacco plants can be used to produce human proteins. Goats are producing human blood clotting proteins. And so it's a really interesting concept here of developing human drugs using plants or other animals. The concern here obviously would be the ethics of using animals for these processes, but again, something really, really important for us. Also in the chapter it mentions stem cells. Now we've briefly discussed these earlier, but stem cells are unspecialized, they're undifferentiated precursor cells, so they don't have a function. It's kind of like, and I've mentioned this before, college students with an undecided major. So stem cells have not taken on a job yet. They eventually will. They're totipotent, so they can become any type of cell in the body. Typically, stem cells are going to be found in umbilical cord blood. They're going to be found in bone marrow, which is why it's a very common thing now during pregnancy for the doctor to ask about preserving the cord blood, because potentially we could take those stem cells. The idea behind it is you could take those stem cells, and since they're totipotent and they can become cell types, you could use them to become any type of cell that you might need to develop any type of organ that you might need. So you could use it for therapeutic cloning. For example, you could grow specific tissues to treat spinal cord injuries, to treat burns, to replace failing organs. You need a kidney, you could use your stem cells to develop your own kidney. You could use it to treat degenerative diseases like liver and lung diseases, heart diseases, to treat Alzheimer's and Parkinson's. So these stem cells actually have a lot of uses that we believe we could use them for. Of course, when we talk about stem cells, a lot of people think of cloning. Identical twins basically are human clones, where the embryo we talked about in the last chapter, chapter 7, where the embryo divides once it is formed. We have already cloned a lot of different types of mammals in the lab, like sheep and rats, cattle, monkeys. The nuclear transfer, the process used for cloning, is where you basically take a adult cell nucleus, you fuse it with an egg cell. And so you can see here the process is, here's a body cell. You remove the nucleus from a body cell of the person you want to clone. You transfer the nucleus from the cell of the person to be cloned into this egg cell. Okay, so here would be your egg cell. And then as that egg cell develops, it turns into an embryo. Embryo implants into the uterus of a woman, and then out pops the clone of the man that you started with. Now we actually have performed in 2013 this nuclear transfer with human cells. It actually produced about 200 cells. And so although human cloning in itself has not occurred yet, more than likely something that we will see eventually happen.